Alright, welcome to the first video on the physiological components of memory, or the physiology behind memory formation. This video is specifically going to look at the neuron and its role in memory formation. So we definitely need to know about the structure of a neuron, and I would advise you to draw one here. With the different parts. So we need to know dendrites, which are these branch-like features here. The soma, or the cell body, is this part here and has the nucleus in it. This long tube here is the axon, which is attached to or finishes with the axon terminal, which is here. The myelin sheath is this protective purple covering around the axon, and we have the synapse, which is the gap between this neuron and the next one. We need to know the function of the different parts of the neuron. You might like to create a table to summarize this information. A dendrite is those branch-like features that receive messages from either other neurons or receptor sites. The cell body contains the nucleus, then that ultimately controls the cell and has the DNA in it. The axon is the thin fiber that extends from the cell body down to the axon terminal, and that's what the electrical impulse, or the message, is transported down or transported through to get to the end of the neuron. The myelin sheath is that protective purple stuff around the axon here, and that helps to speed up transmission. If we don't have the myelin sheath, that action potential, or the impulse that travels down the axon, can get lost or not make it to the axon terminal properly. The axon terminal is obviously the end of the axon, and that's where all the neurotransmitters are stored and released from when the electrical impulse gets there. And those neurotransmitters are released into the synapse, and the synapse is that gap between the axon terminal, so this yellow part here, and either the next neuron or the target cell that it's communicating with. Neurotransmitters are a chemical substance that are stored in the axon terminal here, and when that action potential travels down the axon to the axon terminals, these neurotransmitters are released into the synapse and combined with receptors on the target cell at the other end to stimulate a response in that next cell. We need to know about an impulse, or electrical impulse, an action potential, which obviously starts with the dendrites being stimulated. That electrical impulse travels down the axon to the axon terminals, which release the neurotransmitters into the synapse. They cross that synapse, or the gap there, to reach the dendrites of the next neuron or the receptors on the target cell. So we're going to start with the dendrites, down through the cell body, through the axon terminal, to the axon terminals, and the axon terminals release neurotransmitters into the gap, and that stimulates the next cell. We need to know a couple of different types of neurotransmitters that are specifically involved in memory formation. And then there's a nice summary table here that helps. So we've got glutamate is the first one, which plays a crucial role in the structural changes that occur, particularly in the growth and strengthening of synaptic connections. So if we go back to this picture here, synaptic connections are like the interactions between like the axon terminal and like the dendrites of the next cell. And you can see there's lots of dendrites here. So glutamate is going to help with the growth of those areas, so maybe creating more dendrites or more neurotransmitters being released that strengthen those gaps. If there's more dendrites and more neurotransmitters, we've got a stronger connection between neurons, and that makes them more likely to fire again or to communicate again. So it enhances transmission. Dopamine has been found to be involved in attention and voluntary movement. It also contributes to the strengthening of these synaptic connections, and seems to be an ideal level for working memory to function properly. So we need dopamine for our brains to function properly. Acetylcholine has been found to be involved in learning, attention, sleeping, dreaming, and motor control. So it makes sense that it's found to be abnormally low in Alzheimer's disease, and we're going to talk about Alzheimer's disease later on in this video series. And lastly, norepinephrine has been found to play a crucial role in encoding and retention of memories, especially for emotional experiences, can also be called noradrenaline, and affects memory by influencing the activity of the amygdala and the hippocampus, and we're going to look at them later in this series as well. So definitely know these as examples of neurotransmitters involved in memory formation. Lastly, I want to discuss memory traces. So research has suggested that neural activity is the basis of memory formation and storage, and that each memory has a unique pattern or a unique memory trace attached to it. So they're saying that a memory trace is a physical or chemical change that is believed to occur in the brain, so that they can store this information during memory formation. So those physical and chemical changes could include more dendrites, more axon terminals, more neurotransmitters being produced or formed to make a stronger memory trace. And the stronger the memory trace, the easier it is to find that memory that's associated with that trace. This results in a lasting change that will make the neuron more likely to fire again in the future, which makes sense. So if there's a stronger connection, more dendrites, more neurotransmitters, then they're more likely to fire again in the future, and making it a stronger memory, a more long-lasting memory. And that concludes this video.